Let's go ahead and get started. A lot of folks, of course, have already left for Thanksgiving. It's all right. I would be too if I had the choice. So I'm recording this, so you'll be able to download it or look at it if you wish. Today we're going to talk about this paper. This came out earlier this year. It's a Science Advances paper. An aerobic eukaryotic parasite with functional mitochondria that likely lacks a mitochondrial genome. Now there's a lot to unpack in this, and so I'm going to give you more background than what I actually talk about the paper itself. And so there are a variety of things that we have to kind of get up to speed on before we look at the paper. We need to talk about mitochondrial genomes in general. We need to talk about dinoflagellates in particular. We talked a little bit about dinoflagellates earlier, but they're much weirder than you might think. And particularly parasitic dinoflagellates, of which Amoebophyra is one. Phyra, I think it's pronounced. And that's the subject of the paper itself. So let's start with mitochondria and their genomes. Mitochondria, of course, are the descendants of alpha proteobacteria that became permanent symbionts in eukaryotes, endosymbionts. They carry out aerobic respiration. They carry out ATP synthesis. They also carry out the TCA cycle and a few other kind of metabolic reactions as well. And if you're interested in weird genetics, the place to look is mitochondria. Even mundane creatures like Homo sapiens have really weird mitochondrial genetics. And when you look at protists of a wide variety of kinds, you find all kinds of very strange genetic stuff going on. The genetics of the mitochondria in kinetoplasts, the trypanosomes and euglenids, it's just mind-boggling. And everywhere you look, even in yeast, the mitochondrial genetics is really, really weird. And so if you're interested in genetics that doesn't follow the standard form, look at mitochondria. In all mitochondria, most of the original alpha proteobacterial genes are gone from the mitochondrial genome. Some of those genes have just been thrown away and aren't needed by the symbiont anymore. So, presumably the original host of the mitochondria, the original eukaryote, had genes for, alanine synthesis. So did the organelle. They don't both need them, and so the mitochondria threw it away. Many mitochondria don't have a peptidoglycan cell wall anymore, and so all those genes have been thrown away. Most of the rest of the genes that it still needs, are now in the nuclear genome. In some cases, they just use the nuclear gene for that, right? And now that protein is imported into the mitochondria, as well as functioning in the nucleus or cytoplasm. In some cases, like I mentioned already, you just need the metabolite. You don't need the protein anymore. Because you can get the metabolite from the cytoplasm. In other cases, the original alpha proteobacterial gene has been transplanted into the nucleus, and the product of that then is synthesized in the cytoplasm by cytoplasmic ribosomes and imported, and the protein is imported into the mitochondria. So what do some of these look like? And so this is the mitochondrial genome of Reclinomonas americana. We talked about this organism earlier. It's an excavit. It's got a couple of flagella, and it's got a glass shell that looks like a wine glass. And the organism lays inside of it. That's where the reclino comes from Reclinomonas. It's a Jacobid, and these organisms have what for mitochondria are large genomes. In the case of Reclinomonas, it's about 70,000 base pairs. Some of them have some that are a little bigger, up to 80 or 90,000 base pairs. some of them a bit smaller. And the reclinomonas mitochondrial genome includes the ribosomal operons. Let's see if I can find a pointer here. It includes the ribosomal operons. Where are they? I don't know. They're in here somewhere. It encodes all of the tRNAs it needs. It actually includes some of the other RNAs that are important. So I got interested in this organism because they wanted me to find the RNase pRNA, which this mitochondrial genome has. It's an RNA involved in tRNA processing. It encodes the ribosomal proteins. It encodes the proteins it needs for electron transport. The primary function, of course, of mitochondria is electron transport. It encodes ATPases. It encodes its own RNA polymerases, all that stuff. Doesn't encode any metabolism genes. No genes for amino acid biosynthesis or central metabolism or the TCA cycle, even though the organism carries out the TCA cycle, all those proteins come from the nucleus. And all the other genes then that this thing needs well, all the other proteins that it encoded before have either been thrown away. This organism, the mitochondria of the reclinomonas organism doesn't have peptidoglycans, so all those things are gone. All the genes for lipid biosynthesis for its membranes are now the nuclear homologs instead. But all the other proteins it needs, again, it's either using the nuclear homolog or it's using the alpha proteal gene, but that gene is in the nuclear genome now and not in the mitochondrial genome. This is the human mitochondrial genome. Have any of you ever studied in any of your biology classes human mitochondrial genetics? No? That's a shame because it's really, really interesting. This is the whole thing, 16.5 kb, that's it. It encodes for, that's bad English. It encodes, and actually even, well, I won't get into that. It turns out that, formally speaking, you don't encode an RNA because there's no code involved, right? But still people call it that. It encodes the 2 ribosomal RNAs. The 12S RNA is the small subunit RNA. The 16S RNA is not the same as the 16S RNA in bacteria. It is the large subunit RNA. These RNAs are much shorter than in bacteria. And it encodes 13 proteins of the electron transport chain. Cytochrome B, nicotinamide NADH dehydrogenase, the integral membrane proteins of the ATPase, and the COX 1, 2, and 3 genes. It encodes all the tRNA it needs. Here's the valine, leucine, isoleucine, methionine, etc., all the way around. Why these genes? Why have these genes been kept? Well, one thing you notice right away is that the important RNAs are still here. Why? Well, keep in mind that you're making, presumably, ribosomes inside the mitochondria. If you were to encode those RNAs in the nucleus, those RNAs would have to go from the cytoplasm through both membranes of the mitochondria to get into the matrix of the mitochondria. This RNA right here is about 1,600 base pairs long. That's 1,600 negative charges in a ball you've got to push through membranes. That's pretty tough. Even the tRNAs, you're talking about 75, 80 negative charges in this little structure that you have to push through a polar membrane, and that's hard to do. And so RNA genes typically are retained in organelles. There are many cases, by the way, where that's not the case. I'll show you one of those in a minute. These things are the integral membrane proteins of the electron transport chain. And it's the same problem, right? If you were to normally when you synthesize these, they are inserted into the membrane by ribosomes bound to the membrane, and they're secreted directly into place. You can't do that if you're going to make these proteins in the cytoplasm, push them through the outer membrane of the mitochondrion, and then build them into the inner membrane of the mitochondrion where they belong. And so the fact that you would have to separate translation, and they call it secretion when you secrete a protein into a membrane, physically separate those processes makes it difficult. And so the ones that are retained are the proteins that go back and forth across the membrane and would be really difficult to do that with. The mitochondrial genome of I say humans, but this is all mammals. There's only two transcripts, two promoters. There's what's called the heavy chain, heavy strand promoter that transcribes the genes on this strand, and the light strand promoter that transcribes the genes in this way. Here's the origin of replication. This is the non-coding region that in other systems, this is sometimes called the controlling element or controlling region. This is where gene expression is regulated. And so there are two transcripts, one of each strand, and the individual genes in these transcripts are separated from one another by tRNA processing. And so, for example, here, the 16S RNA and the NADH dehydrogenase gene are separated when this tRNA is cut out and processed. That frees this thing so that it can be used as a messenger RNA. And that's how all of these work. The exception being these two right here and this one right there. There's actually two origins of replication here because this thing replicates by rolling circle replication. Everything else the mitochondrion needs is imported from the nucleus cytoplasm. And there are a couple of things. I mean, for example, this thing doesn't make its own leucine. Leucine is made in the cytoplasm, and it's the metabolite that's pumped in. But all the other genes for the electron transport chain, all the other proteins in the electron transport chain, all the other ATPase subunits, all the proteins for the intermediary metabolism and the TCA cycle are translated in the cytoplasm and imported into the mitochondria. And there are special signals on the protein for how that works. Let's shift gears for a moment and talk about the dinoflagellates. And so this image is a typical dinoflagellate. We talked a little bit about these before. These things are alveolates related to paramecium, for example, and apicomplexia. They're called alveolates because they have these flattened bags of membrane called the alveoli all along their cellular membrane. That's where they get their name. And these can be quite complicated in some organisms. Dinoflagellates are typically called algae. Many of them, most of them maybe have chloroplasts. That's what this is here. They have two flagella. One of them goes down this way, and that's the longitudinal flagellum, and the other one is a transverse flagellum that wraps around the cell. These things have cellulose cell walls. They contain this thing here is where they pull in their prey. These things eat bacteria and other algae and that kind of stuff. They do have mitochondria. Here's the nucleus. In these organisms, I'm going to show you the genomes a bit. Most of the genes of the chloroplast are also gone. And so, and this is true generally. Just like mitochondria, chloroplasts lack many of the genes they need to function. And those are encoded in the nucleus instead. This has gone a little further down that road in dinoflagellates. The chloroplast genome is significantly smaller than in many other creatures. And those genes are those genes where they're needed at all are in the nucleus. It's really interesting is that dinoflagellates have lost the chloroplast in many branches of the group. In most of those branches, even though the chloroplast is gone, the genes, the bulk of the genes that are needed for chloroplast function and development are retained in the nuclear genome. This allows them, and because and that includes most of the genes that are required for the chloroplast. What that means is that when they eat an alga, they can suck up all the nutrients. They can hydrolyze and destroy the eukaryotic part of that creature and keep its chloroplast. And it's got most of the genes in the nucleus that it needs to keep that chloroplast running and dividing and functioning. These are called kleptochloroplasts, and it will keep them for as long as there's light and they're of use to the dinoflagellate. And as soon as they're no longer of any use, they'll complete the digestion of the chloroplast and move on. And so they can go back and forth between keeping chloroplast from somebody else and not. And the fact that most of the chloroplast genomes are now in the nucleus means that they can use chloroplast from a wide range of other algae. Free-living dinoflagellates have some of the largest genomes known. And so some of them have genomes that are 100 times larger than the human genome. You know, we're talking about, upwards of 500 million base pairs. Excuse me, 500 billion base pairs in the genome. Really, really, really big genomes. And those genomes are very strange. So here's another one. This is just a different diagrammatic model. And so these dinoflagellates have their genes in 3 compartments. The nuclear compartment, the genes are really interesting. Again, they have enormous genomes. These genomes are not kept bound up and condensed in nucleosomes for the most part, the way other eukaryotes do. There's so much DNA there, it's actually kept in a kind of a naked form in a very highly organized, basically a liquid crystal form. And the genes that are in that liquid crystal part of the nucleus are inactive. Loops of the genome come out of that liquid crystal and are coded by nucleosomes. And those genes are active. And so the only active genes in this thing are the ones that are in these extra, I don't know what you call extra crystalline loops that are nucleosomed. Gene expression of those things. So the genes in the nucleus are organized into operons, unlike other eukaryotes, right? They're big, long arrays of multiple genes. And once those are transcribed, each protein coding sequence is separated by a process called transsplicing. And so a little RNA is spliced into the front of each one of these. These are called splice leaders. And so that's what this is. So you've got these tandem repeated sequencing, and they're separated, polyadenylated, just like other eukaryotic mRNAs. Every mRNA starts with this splice leader sequence, and that's what the ribosome recognizes to bind and start translation. This seems really weird, but it turns out it's more common in the eukaryotic world than you might imagine. Even the worm, C. elegans, does this. This is an animal. The plastid, where the organism has them, also has a genome, but again, most of the genes are now in the nucleus, not in the plastid itself. Those genes that remain are not found on a single chromosome. Instead, this thing divides this genome up into mini circles. And any particular mini circle will have some subset of the genes of the genome. Oftentimes, only 1 or 2 protein coding sequences on each mini circle, even though they have a collection of, I don't know what it is, 50 or 100 different genes. And so note that the single complete genome of the plastid doesn't exist in any physical way. It's all separated into circles. The mitochondrion is even worse. The mitochondrion of dinoflagellates, the genome is completely fragmented, with only 1 or 2 or 3 genes at a time on these linear pieces of DNA. And so what's encoded in most dinoflagellates' mitochondrial genome are the 2 ribosomal RNAs and 3 proteins, COX-1, COX-3, and COB. cytochrome oxidase 1, cytochrome oxidase 3, and cytochrome B, cytochrome oxidase B. And these things are separated by some various inverted repeats that are involved in processing of these things. These RNAs are edited. In other words, the primary transcript doesn't have the correct sequence. There are enzymes that go in there and correct the sequence to the right protein-coding sequence, using little bits of RNA that are encoded elsewhere here as a template to guide that correction. RNA editing is quite common in weird eukaryotes. And so other than these 5 genes, 2 RNAs and 3 proteins, all the other genes required by the mitochondrion are in the nuclear genome. Or they're no longer required. Again, you don't need amino acid biosynthetic genes because those are made in the cytoplasm. You don't need, the TCA cycle is the proteins are encoded in the nucleus and translated in the cytoplasm and brought in. But these genes, so these 2 RNAs, again, those are going to be hard to push through the membrane because they're big RNAs and lots of negative charges. Notice there's no tRNAs encoded in here. All the tRNAs are brought in from the nuclear genome. Likewise, all the other proteins, again, these are integral membrane proteins. These are the hardest proteins to push through membranes, and so those remain. Well, let's shift gears again. We talked about dinoflagellates and their genomes. Let's talk about the dinoflagellate of today. It's a member of the Syndiniales. These are parasitic dinoflagellates. So they are not photosynthetic. They're not algae in the traditional sense. They're parasites. They're intracellular, obligate parasites of a wide variety of creatures. Animals, what we call algae here, ciliates, basically other alveolates. And even other dinoflagellates. This is a dinoflagellate here. I think this is Carenia. This is the dinoflagellate that causes the Florida red tide. You can see it's got a groove for the transverse flagellum, a groove for the longitudinal flagellum, and these things are the parasites. These parasitic dinoflagellates lack plastids. Not only do they lack plastids, but they lack the plastid genes in their nuclear genome. So they've thrown all those genes away. And they're quite small. They typically have small genomes. I'm sure you can imagine. This thing has got a nucleus in there that's got, billions of base pairs in the genome. There's no space for that in these small parasites. The internal form of the parasite is quite large. In fact, it's a multinucleated body that people call a beehive because of their physical appearance. But what's released out into the environment are the individual, they call them spores, but they're still metabolically active and free-swimming. And these things have more or less a kind of a reduced version of the typical dinoflagellate morphology. Let's take a look at that. So this is the life cycle. The spores infect other creatures, in this case another dinoflagellate. The spore goes into the cytoplasm, and then in most cases, but not all, that thing goes into the nucleus. And so most of these things are nuclear parasites. They take over, but they don't divide. The nuclei divide, but the individual cells don't. And so you end up with this large multinucleate structure. The mother cell then dies and breaks open. And what it releases are actually pieces of this beehive that look something like this. And this thing is a free-swimming form that very quickly divides into individual spores, which can swim around then and infect other creatures. This is actually quite a simple life cycle for a dinoflagellate. Dinoflagellates have really amazingly complex life cycles, and probably a lot of these large genomes is required to direct those complex life cycles. So this is what the infective spore looks like. They call these dinospores. And you know, it's got a body, it's got a groove, it's got two flagella, a longitudinal one and a transverse one. It's got a nucleus. So it's more or less typical morphology. This is what they look like in the nuclear infected nuclei. And so this is another dinoflagellate, and this is the beehive in the nucleus. Each one of these is a nucleus of one of these parasites. And they form these elaborate structures. I think beehive is maybe a bit of a misleading term. These aren't so much like beehives, but they are like the inside of a hornet's nest. If you've ever opened up a hornet's nest to look inside, they have these real spiral, complicated structures inside. That's what this looks like to me. This is a small infected cell, and you can see that that beehive structure has this kind of spiral shape, and those are the formed structures that are released into the environment, the so-called worm-like stage. And then those divide into individual dinospores, and the process repeats. All right, so we're ready for the paper itself now. An aerobic eukaryotic parasite with functional mitochondria that likely lacks a mitochondrial genome. It is a genome sequencing paper. So they've sequenced the genome of this organism and try to figure out what's going on. I'll point out that like most eukaryotic genome sequences, this is not a complete closed genome. In the world of microbes, we're used to the notion that the genome's not done until you've closed all the gaps and you have a single circular chromosome or 2 or 3 chromosomes or whatever it is, and you've got every nucleotide in place. This is not the way eukaryotic genome sequences work. This is not the way the human genome sequence is. And the reason for this is that there are parts of these eukaryotic genomes that are just impossible to sequence. The centromeres, big repeats of short sequences. You sequence into it, you sequence out of it, but you don't really know what's going on in the middle of it. Things near the telomeres. You start to get into repeats, and then you end up into these 3-mer repeats that go over and over again, and you have no idea how many there are out there, and you can't clone and sequence them. Many eukaryotes have big arrays of gene multimers, and so, for example, the Xenopus laevis genome, the clawed frog, contains over 10,000 copies of the ribosomal RNA operon in these big arrays. You sequence into it, you sequence out of it, you try to count how many repeats there are, and that's the best you can do. You're not looking at little variations within those arrays because you have no way, when you sequence one of them, you have no way of knowing where it fits into those arrays. This is really typical in eukaryotic genomes. And like I say, the human genome, there's a couple of percent where it's just not sequenced. They get up, I think, 95% of this genome. For a dinoflagellate, it's small. And so the genome of this thing, again, it's an obligate parasite, it's undergone reductive evolution, is only about 88 million base pairs long, so 20 times bigger than E. coli. This is showing you a phylogenetic tree of the dinoflagellates. So these are the typical, they call them the core dinoflagellates. These are the common free-living dinoflagellates. These are the parasitic dinoflagellates. And here's Amoebophyra right there. Serratia is the one we're looking at today. And these are other apicomplexians. Apicomplexians are primarily parasites. They're related to Plasmodium, Toxoplasma, Cryptosporidium. You probably have heard of these things before. And then also the ciliates like, Tetrahymena and Paramecium, etc. Again, this is an incomplete sequence. It's not closed for the usual kinds of reasons. It's 88 million base pairs long. Interestingly, even though this thing is an obligate parasite, it encodes most metabolic genes. So it's got a complete TCA cycle, and it can make all its own amino acids and most of its cofactors, etc. And so it really does, it's not relying on its host for all this stuff that you would expect a parasite to do. Probably the reason for this is that this organism has a very broad host range. And so it doesn't know what it's going to be able to get from its host because it doesn't know who its host is going to be. The genome contains no contigs or clusters of genes that look like plastid or mitochondrial genes. The organism, like the other parasitic Dinoflagellates, does not have a plastid, and it doesn't have plastid genes. It's thrown away all of those chloroplast-related genes. It has functional mitochondria, which I'll show you in a moment, but there is no evidence of mitochondrial genome. And so in a genome sequence like this, you would expect to get the pieces of the mitochondrial genome, because usually those are amplified. There's lots and lots of copies of them. And you would be able to put them together. And because they're fragmented, you would actually put them together into, it would be weird alternatives of various kinds. But you would get the sequences, and they don't see them. So here's a table that shows you a little bit about the genome compared to some other genomes of other parasitic dinoflagellates that they're also working on. And so again, it's 87.7 million bases long compared to some, these are partly along the way. So here's one that's about 1.5 billion, call it 2 billion, 1.1 billion, almost 5 billion. These are the various scaffolds they have, GC content, et cetera, et cetera. So this thing has approximately 20,000 genes in it, protein coding genes. The average length of these is pretty short. This thing has small proteins. 643 amino acids is the average, compared to these things, that are over 1,000, which is more typical of a eukaryotic protein coding gene. It encodes a full suite of tRNAs, and so it is not presumably required to import tRNAs from its host. It's a little confusing, the reason I hesitate, because you have to remember that there's two kind of parasites we're talking about here. We're talking about the mitochondrion of the parasite, and then the parasite and its host. A bunch of stuff about the genome, etc. These things have introns. A majority of the genes, about 3/4 of them have introns in them, and quite a lot of introns, and they show that these introns are spliced. They have RNA data in here, as well as genome data. Now I said that there's no evidence of mitochondrial genome, and so they searched for those 3 protein coding genes in the nuclear genome, and they found one of them. And so the COX-1 gene, or at least 2 small pieces of it, are encoded in the nuclear genome. And so this is just a sequence alignment showing you what COX-1 gene looks like. And here's a piece, and here's a piece. And so there are 2 pieces, 2 of the core domains of this protein are in the nuclear genome, not in a mitochondrial genome. And they know this because it's assembled with all the other genes in the genome, and so it's flanked on either side by all the assembled nuclear genome. And it's clear that these things are expressed. They're in the RNA data, and introns in these pieces are spliced out. In fact, there's introns that are spliced out, and there's a splice leader at the front of it. They could find no evidence of the other 2 protein coding genes that are commonly found in dinoflagellate mitochondrial genomes, COX-3 and Site B. They couldn't find the other bit of COX-1. The N-terminal piece is AWOL. They can't find it. Nor do they find mitochondrial ribosomal RNAs. Now this makes some sense. I mean, if all the proteins of the mitochondria are imported from the nucleus cytoplasm, what's the point of doing translation inside the mitochondria? And if you're not doing translation, you don't need the tRNAs and you don't need the ribosomal RNA or any of that kind of stuff. And in fact, the nuclear genome lacks also the mitochondrial ribosomal proteins, RNA polymerase, all the other stuff you would expect to be absent. And again, they found no evidence of mitochondrial genome sequences. Well, this raises a really interesting question. Other organisms that have lost their mitochondrial genomes, and there are some, have lost functional mitochondria. And the obvious example of this is yeast. You can take yeast and mutagenize it and destroy the mitochondrial genome. And you can still grow them. These yeast grow anaerobically, of course, because they can't carry out respiration, right? And they're called petites because the cells are small. They don't grow very quickly. And so you can make eukaryotes without mitochondria, and there are eukaryotes that don't have mitochondria. And so the question will be, well, if all the genes are gone, maybe they don't have mitochondria, but they do. And so here's a dinospore. This is an EM, it's cross-section. Here's the nucleus. You can see this liquid crystal DNA of the thing in there. There are two mitochondria. This one, they kind of shaved it twice, so this one and this one are all one. And here they've got a cross-section of the other one. These organisms have two mitochondria per dinospore. There's no plastid or no evidence of plasma. Here are alveola along the inside, although they're not very well organized in this dinospore. Here are some Golgi and et cetera. All the normal kind of stuff is here. Here's the basal body of the flagellum. One of the mitochondria is associated with that basal body. That flagellum needs a lot of energy, right? And so, like a lot of flagellates, it's got one of its mitochondria right there providing energy to power that flagellum. This is a phase contrast image of one of these things. Here's the lateral flagellum. The transverse one will be wrapped around it. They've done a stain with a DNA stain. And so this is a fluorescent dye that intercalates in between base pairs to stain DNA. And the nucleus lights up nice and brightly. Here they do a mitochondrial stain. Now this is a really interesting stain. They call it a mito tracker. Obviously a commercial thing, right? What this is a positively charged fluorescent dye. But although it's charged, it's ionic, it's mostly hydrophobic. And the result is that it can go through membranes. And because it's positively charged, if you've got mitochondria with a proton motive force, that's positive outside, negative inside. And so the dye is attracted to the inside of the mitochondria and accumulates there. And the more the mitochondria tries to keep that proton gradient, the more the dye accumulates into the mitochondria. And it's fluorescent, so you can look at it, and here it is. There's two of them. And when you take the phase contrast image, the nuclear stain, the mitochondrial stain, and superimpose them, there you go. And so you've got functional mitochondria. You know they're functional because they've got a proton motive force. And no evidence of any DNA in them. You don't see the mitochondrial stain here with this DNA stain. Well, that's pretty cool. Well, they've got some genes missing, but when they look around at other organisms, there's actually a surprising bit of variation in how the electron transport complexes work in different organisms. Hopefully you've learned a little bit about this in microbial metabolic regulation. What they propose is this. You've got a TCA cycle. That's all going on from proteins encoded in the nucleus and imported. I can't read it from here. You've got what they call an alternative NADH dehydrogenase. This is the one with the COX-1 fragments in it. This goes to complex 2, which is the succinate dehydrogenase, is the major enzyme here. It's involved in the TCA cycle. This is why the TCA cycle happens in the mitochondria, because you need that succinyl dehydrogenase to provide electrons for complex 2. This goes to quinone. Instead of quinone going to the next complexes, there's an alternative oxidase. This is found in Dinoflagellates generally. That oxidizes the water right there, and you've got this little kind of mini electron transport system. And you've got another one here, which is complex 4 and 5. 5, of course, is the ATPase. And you've got lactate dehydrogenase providing electrons to cytochrome C, complex 4 pumping protons and you've got the oxidase here, the cytochrome oxidase making water here, and this proton gradient drives ATPase. And so they think with just the pieces they have that they can assemble a functional electron transport system, even with cytochrome B oxidase gone and the other components of cytochrome oxidase 3 gone. And all of this stuff is encoded in the nucleus. Well, speaking as an evolutionary biologist, this is mind-boggling. So here's that cross-section again. Here's the mitochondria, this one too. But there's no DNA in there. If you think of this thing still as an alpha proteobacterial endosymbiont, the structure of the thing is here, but the genes that make it are here. The genotype and the phenotype are physically separated from each other. Have any of you ever read anything by Richard Dawkins, very famous evolutionary biologist? He argues very strongly that evolution selective pressure acts on genes. That it's selection is actually gene suites that encode different things. And so in this case, if life is about information, then the mitochondrial organism is here, right? All its genes are here. That's where all the selective pressure acts. All the control, the programming that makes a mitochondria is in here. But the structure, the phenotype is over here. That's pretty amazing. And so when you think about, okay, if you've got mitochondrial function, where is selective pressure acting? It's not acting on here, it's acting here. Where's the organism? We think about life in terms of both metabolic function, well, that's here, and information and coding, well, that's here. And so the two parts of this living thing are in separate places. And so one way to think about this is that mitochondria in general are on this journey toward complete fusion with the eukaryote, right? Going back to the way people used to think about organelles as just a piece of the eukaryote. Well, this organism has completed that journey, right? All the genes are intermingled with its host. It has merged entirely with its host and lost its independence completely. And now I think you can consider this in a very reasonable way, just an organelle. It's just part of the host. Same as the Golgi, same as the lysosome, same as everything else, same as the flagellum. Well, wait a minute. That's not so different than you and I, is it? And so this is a human cell. Here's the nucleus. These are mitochondria. Almost all the genes required for these things, for their structure and function, are in the nucleus. And only the tRNAs, 2 ribosomal RNAs, and 14 proteins are encoded here. And so these mitochondria are also well down that journey toward fusion with the nucleus. It's just that it's more extreme. Even the Jacobins, like Reclinomonas, most of the genes are in the nucleus. And only 70 or 80 of them are still in the mitochondria. This raises a really, to me, interesting thing that has to do with sexual reproduction. Sexual reproduction and organelles are intimately linked, if I can say that. Most organisms that where sexual exchange of DNA and reproduction are linked, as they are in humans, right? You have sexual reproduction. Mitochondria are transmitted from what you consider through a maternal inheritance. That is, one of the two sexes takes on the responsibility of providing most of the resources that developing zygote needs. The eggs are quite large, and they contain mitochondria. The sperm are quite small, and their mitochondria are not incorporated into the zygote. And so the female, the egg, is willing to provide most of the resources in exchange for getting a little bit more inheritance, the mitochondrial inheritance. And in organisms where that doesn't happen, there's often battles and incompatibility and all kinds of interesting things between the two organelles. But it's not as straightforward as that, right? Because all you inherit is the mitochondrial genes. You inherit the structure, and you inherit a handful of RNAs and 13 proteins. And that's it. All the other genes required to make new mitochondria and to make these function, you still inherit both paternally and maternally because they're in the nucleus. And so all you inherit by direct maternal inheritance is the mitochondrial genome, not the mitochondria in their entirety. In the sense that most of the structural and functional genes are in the nucleus, and you inherit half of those from your father and half from your mother. In the case of this organism, this Dinoflagellate, all of its mitochondrial genes are inherited both maternally and paternally. Now, it doesn't undergo sexual reproduction in the same way that we do. It does have sexual reproduction, the sexual exchange, but it's not linked to reproduction the way it is in big multicellular creatures. So this may be why these organisms retain those last few genes, because it's the trade-off in sexual reproduction and eggs versus sperm. This is another Dinoflagellate. This is one that's got chloroplast still, as you can see. And this one's infected. Look at that nucleus. That's a beehive. So, your task. Find something, look at the paper, find something interesting to talk about. That's an easy one. Or, Amoebophyra is a really cool organism. Tell us more about it. Just in terms of its life cycle or structure or anything else. Tell us about its, its nuclear genome, how that works. Parasitic dinoflagellates in general are ecologically critical. It's been estimated that in most red tide situations, a third of the red tide dinoflagellates are killed every day by parasitic dinos like this. And so control of red tide breakouts and fish kills is intimately linked by these parasites. Plastid genomics in dinoflagellates is really, really amazing. So with these mini circles and all that kind of stuff. So tell me about that if you want to look something up. What about human mitochondrial genetics? A pretty large fraction of what you think of as birth defects are genes in the mitochondria, not in the nucleus. So tell me about it. There are other mitochondrial genetics. I mentioned kinetoplasts. There's some really, really weird genetics in kinetoplasts. There's weird genetics in fungi, in the mitochondria of fungi, and so forth and so on. You can go anywhere. The genetics of mitochondria in paramecium is well studied, and it's totally bizarre. Human genetics, I mentioned that. Not all mitochondria do oxidative respiration. There's some that have hydrogenase function instead, and there are some who do other kinds of reactions. They're still mitochondria, they're still descended from the same symbiont, but they have very, very different function. Find one and tell me about it. Whatever you want to do. This is the last of the traditional discussion sessions. The next one is just going to be kind of a reflection thing that we do, and it's going to be next Wednesday, a week from Wednesday, 2 weeks from, well, almost 2 weeks from now, a week and a half from now, after Thanksgiving break. We will not have a discussion session 2 weeks from Friday. Of course, we don't have one next Friday on account of Thanksgiving break. All right, have fun with it.